Welcome to microbiology class. In this video, I am going to tell you about microscopes. Microbiology actually started with the invention of microscope. Because microorganisms are very small, you cannot see microorganisms with our naked eye. So if you wanted to see a microorganism which are very small, like maybe 1 micron in diameter, or 1 micron in length, or 2 microns in length. They are very small, smaller than millimeter, 1 by 1000 of a millimeter, that is 1 micron. So bacteria like Staphylococcus, Streptococcus, Escherichia coli, they are having a size of only 1 microns or 5 microns in length or diameter. So those organisms you cannot see with our eye. So if you wanted to see them or if you wanted to study them, you require a magnifying instrument. Okay. So that magnification, enlargement of the image size, that is magnification. So that can be done by a microscope. So the function of microscope is magnification. The first microscope was developed by Antony van Leeuwenhoek, a Dutch scientist. He developed the first microscope. Later, Robert Hooke, he developed a compound microscope, and many other types of microscopes were developed later. Electron microscope, one latest version of a microscope is there, which can magnify up to 1 lakh times. The image will be magnified up to 1 lakh times. So that type of microscope is also there. Ruska, one scientist, he developed that microscope. So to study microorganisms, to see the cells, to see the smaller structures, we have different types of microscopes available now. So microscope is a magnifying instrument. Because we cannot see them, we cannot see these microorganisms, we need a microscope. One more term you will come across is resolving power. The resolving power of our normal human eye is only 200 micrometers, 200 microns. Microns and micrometers are same. It is 1 by 1000 of a millimeter. Okay, 10 to the power of -6 meters, that is 1 micron. So our naked eye can see a structure which is having a size of 200 micrometers. If it is less than that, we cannot see them. So already I told you microorganisms are having less than that size. Bacteria like Staphylococcus is having only a size of 1 microns in diameter. So to see them, we have to enlarge their image size. For that, we require microscope. Different types of microscopes are available based on the type of organism, based on the type of cell, based on your purpose. Okay, there are different types of microscopes are available, like compound microscope, light compound microscope, UV microscope, otherwise known as a fluorescent microscope, dark field microscope, polarization microscope, inverted microscope, okay, then electron microscope. Like that, various types of microscopes are available. So based on your purpose, based on the type of organism you are going to see, based on the type of diagnosis you are going to do, based on that you can select any of these microscope. Commonly used microscope is a light compound microscope. A light compound microscope. There is a simple microscope also. Normally in botany laboratories and zoology laboratories, they will be using the simple microscope. Simple microscope means only one lens will be used for magnification. Only one lens will be available for magnification. That type of microscopes are known as simple microscope. If you go to a watch repairing shop, that watch repairing person will be keeping a lens in his eyes. That is a simple microscope. Okay, or if you are in a botany laboratory to see the structure of stomata, to see the structure of stem, or in a zoology laboratory, the mouth parts of the cockroach. Okay, that type of things, if you wanted to see, we will be using a simple microscope. The magnification power will be less. Okay, the magnification is normally noted as x. 10x means 10 times. 40x means 40 times. 100x means 100 times magnification. Okay, so that is a simple microscope. Now, if you come to compound microscope, they will be using two lenses. One lens will be near the object. Okay, the slide will be there near the object. That lens is called objective lens, and one lens will be nearer to your eye. That lens is called the eyepiece or ocular lens. So you will be seeing the image through two lenses, one objective lens and one eyepiece, that is ocular lens. So the magnification will be the product of these two. For example, if the objective lens is having 10x, your eyepiece is having 10x magnification power, the total magnification you will be getting is 100 times, not 20 times. 10 plus 10 kadayadu, 10, not 10 plus 10, it is 10 into 10. So 100 times magnification you will be getting. Similarly, if you are using a 40x objective and 10x eyepiece, the magnification, the total magnification you will be getting is 400 times. If you are using a 100x objective and a ocular 10x ocular lens, the total magnification you will be getting is 1000 times. So that is the advantage of a compound microscope. So you will be getting a higher magnification when you are using a compound microscope. So in a normal laboratories, in our laboratory also, we will be using a light compound microscope. Light means for illumination of the object, for brightening the object, for lighting the object, we will be using light. Visible light will be used. An electric bulb or a mirror will be used to brighten the object present in the slide. That's why it is known as light compound microscope. Sometimes the ocular lens, if you are seeing one lens will be present, that type of microscope is present, monoocular microscope. Monoocular microscope. So now if you are continuously using monoocular microscope, that will give you some strain to your eye. So to avoid this strain, normally in some research laboratories and other thing, they will be using a binocular microscope. Binocular, bi means two ocular lenses will be present. So that type of microscope is called binocular light compound microscope. Okay, so that is a compound, basic thing of a compound microscope. If you see the structure, if you see the parts of a microscope, you can see mechanical parts will be the mechanical parts, base, a stand, a stage, okay, horseshoe shaped base you can see, then a handle will be present, then a rotating drum will be present where you can see these objective lenses. Okay, so that is mechanical parts. Magnifying parts, if you see, there are objective lenses and eyepiece lenses, that is magnifying parts. Then another part is illumination parts. Illumination parts means light, for giving a proper light for seeing the object under microscope. We have an electric bulb in some laboratories. Nowadays almost all microscopes are coming with electric bulb. Earlier days we had this one mirror will be there. It will be a concave mirror or a plane mirror. Okay, concave mirror or plane mirror will be there. Using that you can get reflected light. And this light will be passing through a structure present above. It is called as condenser. This condenser focus lights into this object. The slide you are keeping on top, on the stage, that light coming from the condenser will be focusing on the slide. So condenser is a focusing structure. It contains prism and convex lenses. So it will condense light into it. And the condenser will have one more structure called diaphragm. Diaphragm you can open and close it. So you can control the intensity of the light. So that is another structure. present in the microscope, condenser, and inside condenser you can see a diaphragm and source of light. Okay, so parts of microscope you can classify into mechanical parts, magnifying parts, and illumination parts. Okay, these are the basic things of almost all microscope. The appearance and the make will be based on the company, manufacturing company. The shape will be somewhat different, but basically almost all microscope will have these structures. Illumination parts will be there, then mechanical parts will be there, magnifying parts will be there. Magnifying parts, ocular lens, then objective lenses. In some microscope there will be more objective. Normally 3 objective lenses will be present. We call them as 10x objective is will be present, 40x will be there, then 100x will be there. 10x is known as low power objective, 40x is known as high power objective, then 100x objective is sometimes known as oil immersion objective. Because when you are focusing under this 100x objective, you have to put a drop of cedar wood oil on the slide. Okay, so to avoid refraction, okay, to avoid refraction, bending of light rays, we will be adding one oil in the slide. Okay, when you are using 100x objective. Okay, so these are the parts of the microscope. These are the parts of microscope. Basically almost all parts. Then now I told you one condenser. There is one type of microscope called dark field microscope. The common microscope is a bright field microscope. When you are observing this under our common microscope, when you are seeing the field or round thing only, you are going to see the bacteria and other structures. That will be bright. But in the case of dark field microscope, the field will be dark, the bacteria, microorganisms will be bright. So under a dark background you can see bright objects. That type of microscope is called dark field microscope. So in that, a special type of condenser will be fixed. Instead of the normal condenser, they will be fixing a condenser known as dark field condenser. Dark field condenser. Okay, so that is one type of microscope, dark field microscope. Okay, our normal microscope is a bright field microscope. Then we have UV microscope or fluorescent microscope. In that, instead of the normal electric bulb, we will be using UV light. Okay, UV light will be used for illumination. When you are using that type of microscope, you have to use the slide, or the object's slide stain preparation. A special stain preparation is needed. A fluorescent stain should be used when you are using the object should be stained with the fluorescent stains. Then only you will be able to see these objects under UV microscope. The objects will be bright, okay, because of the fluorescence. So you can easily identify the microorganisms. Acid-fast staining, there are some fluorescent acid-fast staining methods are available to see Mycobacterium tuberculosis. Okay, that bacteria can be easily identified by this UV microscope. There are some special staining methods are available. So easily you can identify. So that is a fluorescent microscope or UV microscope. Then polarization is there, inverted microscope is there, that different types of microscopes are there. Then another electron microscope, the latest thing is, it is not an optical instrument. Whatever other microscopes, they are optical. There will be a lens will be there used for magnification. But in electron microscope, instead of lenses, they will be using electromagnets. And there is no light. Instead of light, they will be using electron beam. And the images you cannot see with your naked eye. It will be present in a film, or you can use in a computer, you can see that. Only pictures, it will give you pictures. It will give you pictures, pictures of the object. So that pictures you can see it in a computer or you can see it in a photograph. So electron microscope, there are differences between electron microscope and other microscope. Other microscopes are optical ones, but here there is no, in electron microscope there are no lenses. Instead of lenses they are using electromagnets, and instead of light rays they are using electron beam. And you cannot see, you cannot look into electron microscope. You will be getting only pictures of the electromicrograph. Okay, electromicrograph, that only you will be getting. So that pictures you can study and identify the organism. There are two types of electron microscope. One is called transmission electron microscope. The other one is called scanning electron microscope. Okay, transmission electron microscope, TEM, transmission electron microscope. The other one is called as scanning electron microscope, SEM. Okay, scanning electron microscope is more advanced one. It will give you a three-dimensional picture. Okay, a three-dimensional picture of the object. Transmission electron microscope will give you only a two-dimensional picture. Okay, so there are two types of electron microscope, scanning and transmission electron microscope. Okay, so that is about microscopy. Okay, there are different types of microscope. Without microscope, there is no microbiology. So microscope is a very, very important structure in microbiology. Following this, you will see a video. In that video, you can see the demonstration of parts of microscope and how will you focus under microscope. This microscope which is seen in this video is present in America in CDC, Center for Disease Control. There are differences country to country wise and manufacturing, there will be differences, but basic structures are same in our microscope, the microscope which is available in our laboratory and which is present in this video also. Just watch this video and understand what are the parts of common microscope. The microscope which you are going to see is a light, compound, bright field, binocular microscope. Okay, thank you.
The microscope consists of three main sections: the head, the arm, and the base. Each section contains several important components of the microscope. The first section of the microscope is the head. The head contains the oculars and the nose piece, which hold the objective lens. The oculars are the eyepieces of the microscope. They contain lenses that have a magnification of 10x. In most microscopes, they are binocular. You could adjust the eyepieces to match the distance between your eyes, known as interpupillary distance, so that you can see one image. It is important to look at the image under the microscope with both eyes because it improves the field of view. The objectives attached to the nose piece have a magnification of 10x, which is low power, 20 and 40x, which is high dry, and 100x, which is oil immersion. To determine the total magnification of an object you see in the ocular, you multiply the magnifying power of the objective lens, which is either 10x, 40x, or 100x, by the magnification of the ocular lens, which is 10x. Right underneath the objectives is the stage. The stage, with its mechanical clips, hold the slide in place. In order to move the specimen in the field of view, you may need to move the stage right or left or backward or forward. This is accomplished with the stage controls located under the stage. The stage control knobs are located on the side and beneath the stage. The stage must be kept clean and dry. For this reason, it is a good daily maintenance task to clean the stage as well as the objectives when you use a new slide. The slide will drag on the stage if there's any oil or water on the stage. In the bright field microscope, there are several components under the stage that are essential to obtaining a clear picture of the specimen on the slide. First is the condenser under the stage. It is known as the condenser diaphragm. The condenser diaphragm controls the light that illuminates the specimen, passing through the objective to the oculars. By opening and closing the condenser diaphragm, you control the angle of light, thereby controlling the contrast and depth of field. The second component under the stage, important for obtaining a clear image of the specimen, is the field diaphragm. This component is attached to the third major part of the microscope, The base. The leaves of the field diaphragm control the width of the light that reaches the condenser diaphragm. Both the condenser and the field diaphragm are essential components to obtain proper illumination or Koehler illumination in your microscope. Koehler illumination will be discussed later in this module. There are two types of adjustment knobs on the microscope: coarse adjustment and fine adjustment. Both knobs are used to review a slide containing a specimen. The coarse adjustment knob, which is usually on the side of the microscope arm, is utilized to move the stage up and down to bring the specimen into focus. The fine adjustment knob is inside the coarse adjustment knob and is manipulated to bring the object on the slide into sharper focus. This is usually performed under low power.